Okay, so time for is this 3? It's either 3, I think it's part 4 actually. So, part 4, so POMC converting enzyme or Yepsin A, which is another word for it. So very frequently, as I said earlier, um, we have multiple names for the same thing in biochemistry, and it's really annoying because then if you're trying to look up information on one thing, you have to use all the names sometimes to actually have an understanding of what's going on. Regardless, let's continue. So although many studies indicate that PC13 and PC2 are the major convertases, there are aspartyl-like proteases which may be involved in processing POMC in certain circumstances. So what are aspartyl-like proteases? So think of aspartic acid, which is one of the amino acids. So let's look up aspartic protease just to be sure. So aspartic proteases are catalytic type of proteases or enzymes that use an activated water molecule which is bound to one or more aspartate residues. So this is, well, you can't see it because it's, so this is an aspartate amino acid right here. So here is the amino group and here is the acid group. And here is the residue side chain. And the residue side chain is another acid. So it's aspartic acid. But it's usually negatively charged. So this acid usually becomes deprotonated, which makes this acid group become a conjugate base, or it becomes a negatively charged group. And this negatively charged group is able to interact with waters, which can activate the waters to become they can become better nucleophiles themselves when they're activated by this. So this is an aspartic protease or the eukaryotic aspartyl protease in this particular image, but there are aspartyl-like proteases. So I don't know if that means that they have a different active group. So perhaps perhaps there's a family of these things that look very similar, and there might have been a few mutations, so it might not technically fit the definition of aspartyl-like or aspartyl protease, but perhaps it has most of the same functionality. Regardless, that is what they are referring to when they refer to aspartyl-like proteases. And these may be involved in processing POMC in certain circumstances. So, what would those circumstances be? A mammalian aspartyl protease was identified in 1985 called POMC converting enzyme, so PCE. This is immunologically related to Yapsin-1, which processes at paired basic residues in KEX2 deficient cell lines. So, KEX2 is another protease, and Yapsin-1 will take the place, if you will, of KEX2 in KEX2 deficient cell lines that we're doing research in. So what is Yapsin-1? So it says immunologically related, so it probably means that if you have antibodies against one, they would work against the other. Yeah, let's just look at this real quick. So Yapsin-1, also known as yeast aspartic protease, yeast aspartic protease 3. So YAP, yeast aspartic protease. So that's where the name is coming from. And YAP3 gene product. So it's an enzyme that catalyzes the following chemical reaction. So it hydrolyzes various precursor proteins or peptides with arginine, lysine in P1, and commonly arginine or lysine also in P2. So, or position 2. So the P3 amino acid is usually non-polar, but otherwise additional basic amino acids are favorable in both non-prime and prime positions. Excuse me. So this is an aspartic protease, which also looks for positively charged amino acids. So remember, arginine and lysine are both positively charged amino acids, or they're basic amino acids. So in contrast with aspartic acid, which was an acidic residue, or it becomes negatively charged easily, uh, arginines and lysines are the opposite. They are amino, or one of them is guanidinium and lysine is amino, and they are able, or they're basic, and they are able to take up protons out of the solution and become positively charged themselves. So a lot of these proteases actually look for these dibasic residues where you have in position 1 an arginine or lysine, and commonly in position 2 another arginine or lysine, and then in position 3 it's usually nonpolar, but another basic residue could make it much more easily cleaved as well. So yapsin is functioning like a lot of other polybasic proteases. So here, and remember, this is in yeast. So a mammalian aspartyl protease was identified in 1985 called POMC converting enzyme. So this is PCE. So immunologically related to yapsin-1. So this mammalian enzyme looks very similar to yapsin-1. Anyways, this processes at paired basic residues in the KEX2 deficient cell lines. So like yapsin-1, so yapsin-1, as we saw earlier, will go after dibasic cleavage sites. And remember, a lot of these cleavage sites are in POMC. That is specifically the type of cleavage sites that it prefers to use, these dibasic ones. Anyways, which processes at paired basic residues in KEX2 deficient cell lines. So if you have a cell line that is KEX2 deficient, so let's look up KEX2 just to see what exactly we're talking about. So kexin, and it's also a yeast enzyme, and it's a prohormone processing protease, specifically a yeast serine peptidase. So here, yapsin is an aspartic protease, and kexin is a serine protease. So these are two completely different things. However, we frequently find related proteins in mammals and other organisms, and we often will name the mammalian enzymes after their closest relation in yeast or other microorganisms. And the reason why is we've been doing the research on these simple organisms for so long that we have already established names and structures for a lot of them. So when we discover new and novel enzymes and we find out that they look very similar to this other enzyme, then from the microorganism, then we say, this is the microorganism like protein. And so, for instance, with the POMC converting enzyme, we're talking about how it looks similar to the mammalian, to the yapsin-1. So, yeah, that's just, I had to go on that little tangent right there. So, POMC converting enzyme cleaves POMC to give 21 to 23 kilodalton ACTH, 4.5 kilodalton ACTH, and 13 kilodalton ACTH, which is glycosylated. So there's a sugar that's added to it. So PCE has the ability to replace the, it might be PC13. So it has the ability to replace PC13, or at least do some of the same functions as PC13. Continuing, it also cleaves beta-lipotropin to give beta-melanocyte stimulating hormone. The gene for PCE has not been cloned, and therefore no on-site analysis has been undertaken, or in situ analysis has been undertaken. So, We're really going at our own pace. So other processing enzymes involved in generating POMC-derived peptides. So the further processing of POMC after the action of PC13 and PC2 involves multiple stages and many different enzymes. The production of alpha, beta, and gamma melanocyte-stimulating hormone is particularly complex and occurs in the pars intermedia of the pituitary, in rodents at least, and in other tissues such as the arcuate nucleus of the hypothalamus and the skin. So remember, there would be a reason why the skin might want to have the ability to produce alpha, beta, and gamma melanocyte-stimulating hormone, because the melanocytes are in the skin itself. So there's probably a rationale, biologically or evolutionarily relevant rationale to why the skin would want to have control. Anyways, carboxypeptidase E. As stated above, the prohormone convertases process peptides usually as the carboxyl residue, as the carboxyl residue after a single or paired basic amino acid motif. So carboxyl, after cleavage, and here they're saying the single or paired basic amino acid motif. This is the signaling amino acids for the cleavage to happen. And so, as stated above, the prohormone convertases process peptides usually at the carboxyl residue after these basic amino acids. After cleavage, the lysine and/or arginine residues are removed by the carboxypeptidase E, or CPE, also known as carboxypeptidase H, or encephalin convertase. So we have, what, 3 or 4 names for the same freaking enzyme? Yeah, I can see why people get frustrated with biochemistry. Therefore, in the human anterior pituitary, once POMC has been processed by PC13 at the C terminal of ACTH, there is a lysine-arginine pair of amino acids. And remember, both lysine and arginine are positively charged. They're basic amino acids that are then removed by a carboxypeptidase. So after the first processing of the PC13 at the terminal of ACTH. Therefore, in the human anterior, so once POMC has been processed by PC13 at the C terminal of ACTH, there is a lysine-arginine pair of amino acids that are then removed by a carboxypeptidase. So let's go back to that image to see. So here we have ACTH. So here PC13 is going to cleave this area. But after it cleaves this area, there's still these 2 lysine and arginine residues that are at the very end of it. So this is where PC13 will attack. So the carboxypeptidase E is able to come in and remove these, basic residues later. So there could be further processing that takes place. So similarly, in the production of alpha melanocyte-stimulating hormone, CPE plays an important role in removing lysine and/or arginine residues from the carboxy or the C terminus of ACTH 1 through 17. So 1 through 17, there's another cleavage site within ACTH which produces ACTH 1 through 17. And then those lysine and/or arginine residues can, at the very end of that other cleavage site, can be processed by the CPE, and then, which is the carboxypeptidase E. Then there is there is further removal of glycine to generate the 13 amino acid peptide, which is post-translationally modified to generate alpha melanocyte stimulating hormone. And this is described in more detail in section 2. So going back here, see how there is these, let's go to the human. There's a polybasic cleavage site right here within ACTH. And so there is once this gets cleaved, there are still some basic residues that are associated with this. So these still need to be further removed. These basic amino acids need to be further removed for them to get the final alpha melanocyte stimulating hormone. And here, I'm guessing this jagged area indicates that there was a removal of one of the amino acids on the N-terminus as well, except I'm not too familiar. Focus. There we go. So, however, there is more to the function of CPE than just its role in removal of basic amino acid residues, and it may well be that its secondary role is the more important one for POMC processing. So in 1997, Peng Lo's group showed that CPE, also known as carboxypeptidase E, also acts as a prohormone sorting receptor for the regulated secretory pathway. This function is necessary for prohormones to move from the trans-Golgi network into secretory granules. So the importance of this role is indicated by the results of the CPE gene deletion, which highlights the missorting of prohormones. So if you remove the CPE gene, which would then cause the lack of production of carboxypeptidase E, then the hormones will not be processed correctly and not be secreted into the correct locations. So, continuing, PAM amidates the C-terminal of ACTH. So PAM is I believe we went over this earlier, but so amidase, also known as PAM for peptide amidase. So just peptide amidases, which catalyze the hydrolysis of the C-terminal amide bond of, oh, no, that's not what I want. Oh, what I should say, no, this isn't the one I want. 1 second. So 210, I wouldn't be able, so just know that this is a type of enzyme that is able to add an amino, or I shouldn't say an amino, but add a ammonium to an acidic residue to turn it into an amide. So let us look. Well, what I could do is go back to this group, this right here. So here we have an ammonium, and here is an acid group, a carboxylic acid group. So one side, so what amidases can do is it can go from an amide, which is this group right here, it can take water and use that water to remove this NH2 group, and then you get an ammonium, NH3, and you get a acid. But there are some ways to go from the acid into this amide group as well. And this is a type of processing that happens to some peptides to turn them into their bioactive form or more bioactive forms. So PAM amidates the C-terminal of ACTH in the pathway, creating alpha-melanocyte stimulating hormone, but it can also amidate the C-terminal of joining peptide. Excuse me. This process occurs when the POMC-derived peptides are in the secretory granules. It is difficult to find any evidence for a role for ACTH without the subsequent modifications. So this suggests PAM is critical for the generation of alpha-melanocyte stimulating hormone. As the name implies, PAM amidates the C-terminal of peptides after the basic amino acid residue has been cleaved by CPE. So remember, CPE, the carboxypeptidase E, this is removing the basic amino acids such as lysine and arginine. It's removing them from the ends, the C ends of these peptides. And then after those have been removed, PAM can come along and then start amidating it or adding the amine group to those carboxy acids. So, and primarily at glycine, extended peptides such as is found in the processing to alpha-melanocyte stimulating hormone. PAM is found in most large, dense core secreted vesicles and exists as a bifunctional enzyme with a peptidoglycine alpha hydroxylating monooxygenase, or this is also known as PHM domain, which catalyzes the first stage in the process, and a peptidyl hydroxyglycine alpha amidating lyase domain, which catalyzes the second stage. So this was a paragraph for a sentence, so let's break this down. So PAM is found in most large dense core secreted vesicles. So let's look up what a dense core secreted vesicle is. No, I don't want that. Dense core secretory vesicles revealed as dynamic. No. So here, the typical dense core vesicles. So the vesicles are like the bubbles within the cells. So the typical dense core, which I would assume that those are just dense with stuff that's in it. However, this isn't. I'm not too familiar with this. Regardless, these dense core vesicles deliver protein and neuropeptides to the extracellular space. So they are bubbles that are within the cell, but when these bubbles move to the cell membrane, they basically pop and release their contents into the extracellular space. And this is done by the classic regulated secretory pathway. And while there are more numerous smaller vesicles, which are known as synaptic vesicles, which specialize in the storage and delivery of small neurotransmitters. So here they're just contrasting the typical dense core vesicle with the more numerous smaller vesicles. So apparently the dense core vesicles are also a little bit bigger than the typical smaller vesicles. Regardless, we should continue. And these PAMs, these PAM enzymes, are found in most large, dense core secretory vesicles, and they exist as a bifunctional enzyme with a peptidoglycine alpha hydroxylating monooxygenase. So domain, which catalyzes the first stage in the process, and a peptidyl alpha hydroxyglycine alpha amidating lyase. So let's look up what hydroxyglycine is, and glycine is an amino acid. It's one that has only hydrogens as its, what's it called? It only has hydrogens as its residue. So here you can see N-hydroxyglycine. So this N, how do I phrase that? Here's glycine. So this is the amino acids here, amino group, the N, and your acid group. And then the N has a hydroxy group on the end of it. Okay, and this can be done by certain enzymes. So when they're talking about let me go to here, see if we can get a little bit more detail. Oh, yeah, my bad. So that's hydroxyglycine, but that's not normally done by these enzymes. Alpha hydroxyglycine means that the hydroxy is on the alpha carbon. So here you can see you have your amino group, your acid group, and this would normally be two hydrogens coming off of this alpha carbon. So this is the alpha carbon. However, since this is alpha hydroxyglycine, one of those hydrogens was replaced with this hydroxy group. Okay, so this PAM exists as a bifunctional enzyme with the peptidoglycine alpha hydroxylating monooxygenase, which is why they're calling it a monooxygenase, because you're adding one oxygen atom to that peptide. So this is, oh man, so many words. So peptidyl, which means it's a part of a peptide, the peptidyl glycine, come on, focus, which is the glycine on the peptide. There's an alpha hydroxylating monooxygenase, so this usually takes some form of activated oxygen and can add it onto carbons or other biologically relevant atoms. Anyways, there is a domain or a section of this PAM enzyme which has this function, the peptidoglycine alpha hydroxylating monooxygenase, so PHM. So continuing, this catalyzes the first stage in the process, and a peptidyl alpha hydroxyglycine alpha amidating lyase domain, which catalyzes the second stage. So this PAL domain is very similar to this PHM domain in that it is a domain on the PAM enzyme. And so it's called PAL because it takes a peptidyl alpha hydroxyglycine and then it is an alpha amidating lyase, so which catalyzes the second stage. So the PAL domain is attached to the transmembrane domain and a cytosolic domain, so both catalytic units are held at the membrane but project into the lumen of the large dense core secretory vesicles. So this enzyme seems to have multiple catalytic domains, and these catalytic domains are both pointed on the inside of the lumen, or the inside of the vesicle. So I'm going to look this up on Wikipedia just to make sure that I'm not saying. So yeah, here PAM is a multifunctional protein, and it has two enzymatically active domains with catalytic activity. So the peptidyl glycine alpha hydroxylating monooxygenase, the PHM, and the peptidyl alpha hydroxyglycine alpha amidating lyase, which is the PAL. So this is the PHM version. So we have a peptide which has your here's the peptide, and here the CONH. This is the amide group between the rest of the peptide, and this part is the glycine, so the NHCH2CHO2 minus. This is the glycine group. And so this PHM of the PAM will take an oxygen, and then it adds two acidic protons, and then it is able to produce a water. One of the oxygens of water is coming from the oxygen, and then this OH over here was added to the alpha carbon of the glycine, which is at the very end of the peptide. So, and then the PAL subunit takes the same peptide that's already been processed, and so this peptide ends up taking that how do I phrase that? Ends up taking this peptide right here, that has the OH on the alpha carbon, and then it uses it to complete the removal of the glycine residue, but it does it in a way that leaves the amino group that was originally a part of the glycine. It leaves that amino group as the amide group on the full peptide. So this eliminated product is glyoxylate. So here's glyoxylate. So this, let's compare glyoxylate with, well, I could compare it with glycine, but so this was originally part of a glycine. So glycine, rather than having this O double bonded oxygen here, it originally had two hydrogens, and then it had this amino group that was attached to it as well. But then the monooxygenase came along and added the hydroxy group here, and then another part of the enzyme ended up using that oxygen, this oxygen, to kick off the amino group, or the amide group. And so this leaves the amide group on the rest of the peptide. So, yeah, that was quite a bit to go through. Anyways, the PAL domain is attached to a transmembrane domain. Oh, and we already said that. So the secretory granule endoproteases cleave the two domains from the membrane so that they exist in the lumen of the granules. However, there's also a naturally occurring soluble form called PHM4, made up only of the PAL domain. And remember, the PAL domain is the second part of the chemical reactions. And these are generated by alternative splicing. So this can just happen within the cells normally. Anyways, there is also evidence that PAM alters the organization of the actin cytoskeleton, which is important in the release of secretory vesicles from cells. So actin is one of the proteins that basically forms web-like structures in the cell, and it moves stuff around in the cell. And there's also evidence that PAM alters the organization of these actin filaments. So the role of PAM in POMC processing in the hypothalamus has received little attention, and there are currently no reports of mutations in humans that have resulted in obesity. If PAM is critical in the generation of alpha melanocyte stimulating hormone, in subjects carrying deleterious inactivating mutations, it may be there is a degree of redundancy in the system with other enzymes undertaking similar amidating activity to compensate. So remember, if rodents, and I presume people as well, if they do not have the ability to produce alpha melanocyte stimulating hormone, and part of that could have been done from a PC2 mutation. If they don't have this, then obesity is one of the typical side effects or pathologies. So if PAM is critical, well, here we're saying PAM is not critical for this. We just know that it serves a typical functional role, but in there are no reports of mutations in humans that have resulted in obesity from this PAM. So that would indicate that there is another way to, if PAM gets deleted somehow, there is redundancy in the system. There's another way for these processes to continue. So I think we're at 37 minutes. So with that, I'm going to head on off. So I'll see you next time.